of Dr. Victor Rao Vikhe Patil Foundation's Medical College and Hospital at Ahilyabagar, Maharashtra, India, for inviting me as a resource faculty in this international CME on updates on immunophysiology of diseases. This is an online lecture. Immunity, or the defense system of the body, is an intriguing topic. Tremendous research has happened at molecular level and disease presentation and treatment level. We often see a wide spectrum of diseases where low immunity in a person leads to small diseases like common cold to an increasing number of autoimmune disorders. This lecture intends to present the basic normal mechanisms of immunity of our body. This is the outline of the lecture. We'll start with definition of immunity and slowly go into details of types of immunity and inflammation and immune tolerance. Immunity is an ability to resist almost all types of organisms or toxins that tend to damage body tissues and organs. Without immune response, we would not be alive. Small triggers happen in everyday life. For example, exposure to allergens like strong perfumes or pollens, or exposure to organisms like influenza virus or many other bacteria. But we don't often fall sick. We have strength to resist. This is immunity. Immunity is generally classified into two types: innate immunity and acquired immunity. Innate immunities are ancient natural mechanism to prevent invasion of organisms in the body. We also develop immunity with consequent exposure, which is called acquired immunity or adaptive immunity. Acquired immunity is of two types: humoral immunity and cell-mediated immunity. Science says that around 450 million years ago, a genetic event led to acquired immunity. There was probably insertion of transposon into the genome in a way which made possible for generations of the immense repertoire of T-cell response and antibodies that can be produced by the body. Transposon is a class of genetic element that can jump to different locations within a genome. Let us delve into details of these both types of immune responses. Innate immunity is our first line of defense against anything foreign. It defends the body against pathogens and it provides an immediate, though with non-specific response to pathogens. This form of defense does not provide long-lasting protection, but it is an essential barrier to infection. These natural mechanisms include skin barrier, saliva, tears, cytokines, complement system, lysosomes, bacterial flora in the gut, and numerous cells including neutrophils, basophils, eosinophils, monocytes, macrophages, reticuloendothelial system, natural killer cells, epithelial cells, red blood cells, and platelets. Physical and chemical barriers are the first elements of innate immune response. It prevents pathogens from entering the body. Skin is the best physical barrier with a very tough outer layer of dead cells and antimicrobial peptides. Mucous membrane that line the respiratory system, gastrointestinal system, and urogenital tracts, trapping many pathogens in mucus. Secretions like saliva, tears, gastric acid contains enzymes such as lysozymes that can break into bacterial walls. Various cell types play a critical role in the innate immune response, like phagocytes, including macrophages, neutrophils, dendritic cells. They engulf and digest pathogens through phagocytosis. Natural killer cells can recognize and kill infected or cancerous cells by inducing apoptosis, that is programmed cell death. Mast cells and basophils release histamine and other chemicals during inflammatory response, increasing blood flow and recruiting other immune cells to the site of infection. There are soluble mediators which include basic polypeptides which react with and they inactivate certain types of gram-positive organisms. Cytokines, these are signaling molecules such as interleukins and interferons, which coordinate the immune response by promoting cell communication and activating immune cells. Complement systems are a group of proteins that, when activated, can directly kill pathogens or facilitate phagocyte uptake through opsonization. Acute phase proteins such as C-reactive protein, which increase in response to infection, we have seen this in COVID time, and inflammation, they enhance immune response. Innate immunity, it makes the human body resistant to diseases such as some paralytic viral infections of animals, hog cholera, cattle plague, or distemper. A viral disease that kills a large percentage of dogs and that become afflicted with it. Likewise, many animals are resistant or even immune to many human diseases such as poliomyelitis, mumps, human cholera, measles, and syphilis. So innate immunity is naturally present in all. It's not only humans but also in animals, and they protect body against many organisms which can invade into the system. For those who are interested, here is the ancient evolutionary origin of the innate immunity since, imagine, 900 million years ago. So much of research has gone into this. Comparative physiology like this is part of study of physiology, I would say. So I personally love this figure. This is from Kelly's textbook of rheumatology. See, the innate immunity is a generalized immunity, but body also needs a specific response. So, human body also has ability to develop extremely powerful specific immunity against individual invading agents such as lethal bacteria. Against every bacteria we have a specific immunity. Viruses, toxins, and even foreign tissues from other animals, example is transplanted organs. This ability is called acquired immunity. Here are certain characteristics of this acquired immunity. Acquired immunity is a product of body's lymphocytes, T and B, in people who have a genetic lack of lymphocytes or whose lymphocytes have been destroyed by either radiations or chemicals. No acquired immunity can develop. Both T and B lymphocytes are derived originally in the embryo from pluripotent hemopoietic stem cells that form common lymphoid progenitor cells and then lymphoblast, and then they stay in lymphoid tissues as T and B cells. This type of immunity provides a specific response to pathogens, and it also retains memory of the past infections, allowing for a more rapid, robust and a strong response upon subsequent exposure. We know about this primary and secondary response. So, acquired immunity is characterized by the ability to recognize very specific antigens and offer a very diverse response. How do T and B lymphocytes become so specific? Once lymphocytes are formed from common lymphoid progenitor cells, they get pre-processed separately and then eventually end up in lymphoid tissue here. The B lymphocytes are pre-processed in liver during mid-fetal life and during late fetal life, but after birth they are processed in the bone marrow. This population of cells, that is B lymphocyte, was first discovered in birds which have a special processing organ called bursa of Fabricius, and for this reason these lymphocytes are called B lymphocytes to designate this role of bursa. The lymphoid progenitor cells, which are destined to be T lymphocytes or activated T lymphocytes, they first migrate to thymus here and are pre-processed here. Hence, they are called T lymphocytes, designating T for thymus. Please understand that each of these lymphocytes is capable of forming only one type of antibody or one type of specifically activated T cells with a single type of specificity, and only specific antigen can activate it. This is very precisely managed. The question may arise that how can we develop specificity against millions of organisms? For each functional T and B lymphocyte, the gene structure codes for only one single antigen specificity. These mature cells then become highly specific T and B cells that spread to and then they go all over the body and populate the lymphoid tissue. Now there are gene segments. The combination of these gene segments create millions, literally millions of antigenic specificity. This is an incredible mechanism. How does this all happen? Different lymphocytes that are capable of forming one specific antibody or T cell are called a clone of lymphocytes. So there is B clone and T cell clone. Macrophages, they line lymph nodes, spleen and other lymphoid tissues. They lie in opposition to many of the lymphocytes and lymph nodes. Most invading organisms are first phagocytized and partially digested by the macrophages. Their antigenic products are liberated into macrophage cytosol inside the cytoplasm. Macrophages then pass these antigens to lymphocytes literally by cell-to-cell contact. They personally go and deliver it. This leads to activation of specified lymphocytic clones. Macrophages also secrete interleukin-1, which promotes further growth and reproduction of these specific lymphocytes. Most antigens activate T and B lymphocytes at the same time. Let's look at the humoral immunity, that is B cell immunity mechanism first. Before exposure to a specific antigen, the clones of B lymphocytes remain dormant. They are quiet in the lymphoid tissue. After exposure to antigen, macrophage activates B cell clone and also T cell clone. T cell clone forms helper T cells, interleukin from macrophage, and these helper T cells together contribute to extreme activation of B lymphocyte. B lymphocytes specific for that given antigen immediately enlarge and take the appearance of lymphoblast. Some of the lymphoblasts differentiate to form plasmablast, which are precursors of plasma cells. These are plasma cells. In the plasmablast, the cytoplasm expands and the rough endoplasmic reticulum proliferates greatly. The plasmablast then start to divide at a rate of about once every 10 hours for about 9 divisions, giving total population of 500 cells for each original plasmablast, and this happens in 4 days. The mature plasma cells then produces gamma globulin antibodies, that is IgGs and IgMs and all that, at a very rapid rate of about 200 molecules per second per plasma cell. These antibodies are secreted into. Finally, the plasma cell dies with exhaustion. Some lymphoblasts form a new B lymphocyte similar to those of the original clone with that specific antigen. They are called memory cells. This way, B cell population of the specifically activated clone significantly increases. These new B lymphocytes are added to the original lymphocyte clone of the same antigenic specificity. Memory B cells functioning this way also explain the difference in primary and secondary response properly. The secondary response is always quicker and stronger. The example of these long-lived plasma cells is that It provides a lifelong immunity against infectious diseases like measles and smallpox. Now coming to T cell. On exposure to the proper antigen presented by the macrophage, the T lymphocytes of the specific clone proliferate and release large numbers of activated specially reacting T cells. The principal difference between B and T cells is that B cells release antibodies, but T cells get activated on their own and they function to their specificity. These T cells then pass into circulation and are distributed throughout the capillary walls into the tissue spaces, back into the lymph and then blood again, and circulate again and again throughout the body, sometimes lasting for months or even years. T lymphocyte memory cells are also formed in the same way as B lymphocytes, and they are added to the original clone. T cell responses are extremely antigen specific, like antibody responses of the B cells. B lymphocytes recognize intact antigen, but T lymphocytes respond to antigens only when they are bound to the specific molecules called MHC proteins or major histocompatibility proteins, which are present on the surface of the antigen presenting cells or APCs. They are in the lymphoid tissues. Three major types of these antigen presenting cells are macrophage, B lymphocyte and dendritic cells. The dendritic cells are the most potent and powerful. APCs are located throughout the body. The APCs communicate with T cells through major histocompatibility protein. These MHC proteins bind with peptide fragment of antigen protein that are degraded inside AP cells and then transport them to the cell surface. There are two types of MHC proteins. One is MHC1 protein, which presents antigen to the cytotoxic cell, and MHC2 protein, which presents antigen to the helper cell. One fact is that, please understand, adaptive immunity system can prepare a response to pathogen only after the pathogen has generated signals via PRR, which is pattern recognition receptor of the innate immunity system. So innate immunity is required to initiate acquired immunity. The details of how this happens is beyond the scope of this lecture. The B lymphocytes have greater diversity of response than T lymphocytes, and they form millions and millions of varieties of antibodies with different specific reactivities. This is how the MHC protein presents antigen to T cells from the AP cells. So these are antigen presenting cells. This is MHC protein. From the cell surface, then they will present antigen to the T cell and then they get activated. Antibodies secreted by B lymphocytes are gamma globulins and are called as immunoglobulins or Igs. Each antibody is specific for a particular antigen, and this characteristic is a result of unique structural organization of amino acids in the variable portion. This is a variable portion of heavy and light chains of the antibody. The variable portion attaches specifically to a particular type of antigen. So these are the sites where antigens bind. The constant portion determines other properties of antibody like diffusivity in the tissue, adherence to specific structure, or attachment to complement complex, etc. There are 5 classes of immunoglobulins: IgG, IgM, IgA, IgD, and IgE. IgG constitutes around 75% of the antibodies. Ig is small percentage and it is effective against allergy. How do these antibodies function? Antibodies act mainly in 2 ways to protect the body against invading agents. One is by direct attack on the invader, and the second one is by the activation of the complement system, which has got multiple other ways to act. The direct action includes processes like agglutination, in which multiple large particles with antigens on their surface, that is red cells, bacteria, etc., they are bound together and they get clumped together. Precipitation, in which the molecular complex of soluble antigen and antibody uh, becomes so large that it is made insoluble and it gets precipitated. Neutralization, in which the antibodies cover the toxic sites on antigenic agent. And fourth is lysis, in which some potent antibodies are occasionally capable of directly attacking membranes of cellular agents and they rupture the agent. These direct actions of antibodies are often not very strong to play a major role in protecting the body against invader. So the need for amplified effects through the complement system is very essential. So here is a complement system. Complement is a collective term for about 20 proteins and many of which are enzyme precursors. This is a classical pathway and it is initiated by an antigen-antibody reaction which uncovers or activates a specific reactive site on a constant portion of the antibody. We saw the constant portion. This binds directly with C1 molecule of the complement system. This sets into motion a cascade of sequential reaction as shown in the figure. The C1 enzyme activates successively increasing quantities of other enzymes in the cascade. This is how a small effect of initiation leads to a much amplified and a major powerful effect. Some of the important effects of this cascade reaction are: one is opsonization and phagocytosis. C3b strongly activates phagocytes by neutrophils and macrophages, and these cells engulf bacteria to which the antigen-antibody complexes are attached. Second is lysis. The membrane attacks complex, they are also called as cytotoxic complex, and it is a combination of multiple complement factors, that is C5, B, 6, 7, 8 and 9. This membrane attack complex inserts itself into the lipid bilayer of the cell membrane, creating pores that are permeable to ions, and this causes osmotic rupture of the cell membrane of bacteria or any other invading organism. This is the most powerful mechanism. Third is agglutination, the complement product that changes the surface of invading organism, causing them to adhere to each other. Fourth is neutralization of viruses. Fifth is chemotaxis. The fragments of C5a initiates chemotaxis or migration of neutrophils and macrophages to the area adjacent to the antigenic effect. The WBCs are moved towards where there is an injury. Activation of mast cells and basophil fragments, that is C3, C4a and C5a, activate mast cells and basophils, causing them to release histamine and heparin and many other substances into local fluids. These substances cause increased local blood flow, increased leakage of fluid and plasma protein into the tissue, and other local tissue reactions which help inactivate or immobilize the antigenic agent. These same factors play also a major role in inflammation. The role of inflammation is to prevent the movement of invading organism through the tissue. This is acute inflammation. The chronic inflammation is completely different story. Acute inflammation is primitive, protective response, and chronic inflammation involves failure of mechanism designed to stop the acute response. So just to summarize, mechanism of antibody reaction is through direct action or activating complement system, which comprises of 20 proteins. It gives much stronger protection, it gives much stronger action against invading organisms. There are three types of T cells: helper T cells, cytotoxic T cells, and regulatory T cells. T helper cells help in functions of immune system in many ways. They serve as a major regulator of virtually all immune functions. They form lymphokines that act on other cells of immune system on bone marrow cells. In the absence of lymphokines from the T helper cells, the remainder of the immune system is almost paralyzed. It cannot function well. The human immunodeficiency virus, that is HIV, destroys these T helper cells and causes AIDS. What are the functions of T cells? Lymphokines and interleukin-2 stimulates growth and proliferation of cytotoxic and regulatory T cells. Almost all interleukins, but especially IL-4, 5, 6, they participate in B cell activation and growth and differentiation to form plasma cells and antibodies. T helper cells slow down and stop the migration or chemotaxis of macrophages into inflamed tissue. This increases accumulation of macrophages in the required area. Helper T cells also activate the macrophages to cause much more efficient phagocytosis, allowing destruction of many more numbers of bacteria or other invaders. Interleukin-2 has a direct positive feedback in stimulating activation of T helper cells, so it is a positive feedback among each other. Cytotoxic T cell is a direct attack cell that is capable of killing microorganisms, but it can also kill body's own cells too. The receptor proteins on the surface of CD8 cytotoxic cells cause them to bind tightly to the organism or cells that contain appropriate antigen specific antigen. These these cells they secrete perforin and punch a hole into the cell wall of the organism. Then they pour cytotoxic substance directly into the attack cells, and that's how then they kill the attack cell. Cytotoxic cells also play an important role in destroying cancer cells, heart transplant cells, or other types of cells that are foreign to a person's own. So sometimes they are helpful, sometimes they are not helpful. Third, regulatory T cells. Very less is known about them, but they are capable of suppressing the functions of both cytotoxic and helper T cells. These suppressor functions of CD4 regulatory T cells is believed to prevent the cytotoxic cells from causing excessive immune reaction that might damage person's own body tissue. This is an important role in limiting the ability of immune system to attack person's own body tissues called as immune tolerance. We'll just discuss this in the next slide. One unavoidable consequence of creating a diverse types of antigen receptor is the risk of expressing receptor reactive to self tissues. Therefore, a host must employ a mechanism to protect itself against B and T cells. The immune mechanism normally recognizes body's own tissue as being different, distinctive from viruses, bacteria, and other invading organisms or toxins. But if this fails, the person's immunity system will form a few antibodies or activated T cells against his or her own antigens and will damage our own tissue. We call them as autoimmune disorders. So what is the mechanism of this self-protection and where does it happen? These protective measures are collectively known as immune tolerance. There are two types, central and peripheral. Central tolerance or the tolerance itself is a feature of adaptive immunity, is not a feature of innate immunity. For B cells, the central tolerance occurs primarily in the bone marrow, and for T cells, it happens in thymus. This is called pre-processing. Although All lymphocytes in the body originate from lymphocyte-committed stem cells of the embryo, as we have seen. These stem cells are not capable of forming activated T cells or antibodies directly. They must be differentiated with an appropriate processing in respective areas. The T lymphocytes, after origination in the bone marrow, first migrate to the thymus. We saw that here they divide rapidly and at the same time develop extreme diversity for reacting against the thousands of different specific antigens. And then these pre-processed T cells leave thymus and spread all through the body via circulation, then they get lodged into lymphoid tissue. But before they are released after pre-processing from the thymus makes it certain that the T cells will not react against proteins or antigens that are present in our own body tissues. Otherwise, these T cells will be lethal to our body only in few days. So what happens then? The thymus first exposes T cells to almost all specific self-antigens from the body's own tissue. If a T lymphocyte reacts strongly to these self-antigens or has no reaction at all, it is destroyed or phagocytized instead of being released from thymus, so it doesn't enter into the circulation. These cells undergo apoptosis. In other cases, T cells with reactive, autoreactive receptors, they are allowed to develop, but these T cells are released as regulatory T cells, which adopt inhibitory function, which suppresses immune response in the periphery, which we just saw by inhibiting cytotoxic T cells. So, finally, the T cells which are released from thymus outside, they react only against the antigens other than the body, that is outside source, like bacteria and etc., or even a transplanted tissue from another person. Most of the pre-processing of T lymphocytes in the thymus occurs shortly after the birth of baby or for a few months after the birth. In humans, B lymphocytes are pre-processed in liver during mid-fetal life and in the bone marrow during late fetal life and after birth. So this was central tolerance. The peripheral tolerance is, although central tolerance is an effective process, but there could be some cells which are emerging through lymphocyte development which can still react to self-antigens. Hence, there are mechanisms in place to guard against destruction of these self-tissues after T and B lymphocyte cellular maturation is complete. This is called peripheral tolerance, and this involves multiple signaling pathways and processing mechanisms. Regulatory T cells are also involved in the peripheral tolerance. Dear all audience, it will be inappropriate to discuss the role of immunity without discussing the roles of certain environmental factors and some aspects of our daily life which influence our immune response, and many times this leads to chronic inflammation. Chronic inflammation is a cause of many diseases. Some such culprits behind most of the cases of chronic inflammation include low level of physical activity. And see, this is what we have done to ourselves. Chronic stress, role of corticosteroids and other stress hormones like thyroid and adrenaline, and psychoneuroimmunology, high BMI, high visceral fat, imbalance of healthy and unhealthy microbes in our gut, regular consumption of inflammatory food like food high in trans fat and sugars and salt, disrupted sleep and circadian rhythm, thanks to social media, exposure to toxins, viruses, and we don't have a defense system, and using tobacco products and alcohol. List is endless, and there are so many factors which really influence and modify our immune response. It is our duty to maintain our immune response stronger and intact. At the end, I thank you all. It was nice to discuss the basics of physiology of immune response. I once again thank Professor Nigute, Joint Secretary and HOD Physiology, Dr. Avari, Deputy Dean, Organizing Secretary of the CME and Head of the Department Microbiology, and Dr. Vittal Rao K. Patil Medical College and Hospital for giving me this opportunity to speak. I also thank you all, our audience, for being really patiently listening to me. So thank you so much.